The only sign that they have hepatitis is that they have hepatitis virus RNA in their circulation. Otherwise, they have normal liver histology by all means of pathological examinations, ultrasound, and everything. They have normal hepatic enzymes. They have constantly low viremia, and they have no extrahepatic manifestation. The most important is that they have almost none or at least reduced core antigen level. The core antigen of hepatitis C virus is the major pathogenic protein of the virus that has been shown to be involved in the hepatitis, steatosis, cirrhosis, and also recently the transgenic mice with core antigen could develop hepatocellular carcinoma within about 45 days of their lives. So which means that the core antigen level is a real pathogenic indicator, or the real indicator, biological indicator for the hepatitis C infection. So what are the factors that may influence the severity of the HCV-related liver disease? These factors might be viral, host, or cellular. Viral, by definition, is those mutations that happen due to the lack of fidelity of the HCV RNA-dependent RNA polymerase, and the mutations, especially in those genes that are known to be phylogenetically conserved among all the isolates of hepatitis C. Any mutation within these highly conserved regions may have influence on the life cycle of the virus. The host means this endocrine derived or driven differentiation between males and females, the immune composition or the immune makeup of the patient. Cellular means those transactive, lots of transactive factors, nuclear transactive factors that interact with the viral genome and make up the entire phenotype of the HCV infection. So we will focus in this talk on the viral factors, and we will try to derive the important sites of, or the important sequence within the viral genome that will help us to design a suitable antisense molecule for as an alternative therapy. So this is the HCV genome. It is tentative. The HCV genome is a single strand RNA of a 9.6 base, kilo base, and the genes within the genome encode for either structural or non-structural proteins. The structural proteins include the core and the envelope glycoproteins. The non-structural, the genes that code for the non-structural proteins mainly encode for the group of enzymes related to the viral replication, serine protease, helicase, and RNA-dependent RNA polymerase. Due to the lack of fidelity in this enzyme, lots of mutations are encountered, particularly in the envelope and the core. And this is for reasons for the virus to escape from the immune stress of the host. However, there are very highly conserved regions, which are the regions coding for the viral replicating enzymes, and also both the 5' and the 3' untranslated regions. May I have another pointer, please? So these 5' and the 3' untranslated regions are characterized by that they are very highly conserved among all the reported quasi-species of hepatitis C. And the reason of the conservation may be related to that the structure is highly structured, and it contains lots of secondary structure with stem loops around here, and these stem loops triggers the potential binding with the transactive factors. Oh, so this is fine. This is fine. This is fine.
He likes fat steak.
Yes.
So this is a magnification of the 5' upstream region, which is also called the iris region, the internal ribosomal entry site, because it is the major region of the virus that is related to the initiation of viral protein translation at the host ribosomes. The most important of this secondary structure is the initiating methionine, and this is the initiation of the core gene. So the AUG start codon is here. And when we go upstream towards the 5' and the entire length of this is 340 bases. And this secondary structure includes a 3D structure, or we call it the third complex structure, and it can be termed as 3A, 3B, 3C, and 3D. And this 3D is one of the most important secondary structures of the iris region of hepatitis C genome. The S1 nuclease protection assay has revealed that this is the 3A, 3B, 3C, and 3D. So the eukaryotic initiation factor 3 may bind to these loop structures, and it regulates the initiation of translation, and also it regulates the expression of viral proteins. The 40S ribosomal subunit and the 5S ribosomal subunit will bind to the loop structure at the 3D complex. And this triplet G around here so the 40S ribosomal subunit protein and the 5S ribosomal subunit protein will bind to the loop structure around here. And it is very important to have a triplet G at the apex of the loop structure. Site-directed mutagenesis studies have revealed that elimination of any of these Gs will dramatically knock out the binding of 40S subunit protein to the loop structure at the 3D stem loop. And eventually it will affect dramatically the translation of the polyprotein precursor of the virus, starting from the initiating methionine, and the viral proteins will be dramatically reduced. So going back to the patients that I have described in the beginning, who are termed as healthy carriers, these patients, we tried to analyze why they are healthy. Probably their immune system was different from the immune system of the patients with aggressive liver disease. And probably the viral quasi-species infecting these people have dramatic mutations that affect the viral replication. So we decided to analyze the sequence at the upstream of the 5' upstream region, and particularly we concentrated on the 3D stem loop structure in our patient. So this is the triplet G that is very important for binding to the 40S subunit. And by analyzing some of these patients, we found some of these patients encountering mutations, just single or double mutations somewhere. So this is an example of these mutations. a GC has been converted into AG. And what happened is that the 6 loop, the 6 nucleotide loop has been, by the electronic flow, has been changed into an octa loop or octanucleotide loop with a partial shuffling of the triplet G to be not in the center or at the apex of the stem loop structure, which with a possibility of failure of binding of the ribosomal subunit and the consequences of reduced viral replication and reduced gene expression. So this is the type of dinucleotide mutation that we found in one of the healthy carrier patients. And we have analyzed the stem loop structure in a number of these patients.